How to address the risks of gene drives altering ecosystems. Understanding gene drives and their purpose. Gene drives are genetic engineering tools designed to rapidly spread specific traits through wild populations. They have the potential to eliminate disease-carrying mosquitoes, control invasive species, and enhance agriculture. However, their ability to alter entire ecosystems raises concerns about unintended consequences and long-term ecological effects. Potential ecological risks of gene drives. One major risk of gene drives is their unpredictability in natural environments. Once released, these modified genes could spread uncontrollably, leading to unintended harm to native species. For example, altering insect populations could disrupt food chains, affecting birds, reptiles, and other dependent animals. Preventing unintended spread of modified genes. To minimize risks, scientists are developing containment strategies, such as reversible gene drives, that allow genetic modifications to be undone. Additionally, laboratory testing and controlled field trials are essential before any large-scale release. These measures help ensure that gene drives function as intended without causing ecological damage. Ethical considerations in gene drive deployment. Altering ecosystems through gene drives raises ethical concerns, particularly regarding human intervention in nature. Who decides which species should be modified or eliminated? Engaging local communities, policymakers, and environmental experts is essential to ensure responsible decision-making and address moral dilemmas. Regulatory frameworks for safe application. Governments and international bodies must establish strict regulatory frameworks to oversee gene drive research and deployment. Policies should include risk assessments, public consultations, and guidelines for monitoring and controlling gene drive organisms. Transparency and collaboration between nations are key to ensuring safety. Mitigating risks through targeted gene drive design. Scientists are working on more precise gene drive technologies, such as precision-guided sterile insect techniques, which limit modifications to specific populations. These approaches reduce the risk of unwanted spread and provide better control over genetic alterations in wild species. Monitoring and long-term impact studies. Continuous monitoring of ecosystems after gene drive deployment is crucial. Researchers must track how altered species interact with their environment over multiple generations. Long-term studies help identify potential problems early, allowing for corrective actions if negative effects arise. Balancing benefits with environmental caution. While gene drives offer promising solutions to global challenges like malaria and invasive species control, they must be approached with caution. Striking a balance between innovation and ecological preservation is essential to harness their benefits without harming biodiversity. Public awareness and global cooperation. Public understanding of gene drives is limited, leading to skepticism and fear. Educating communities about both the potential benefits and risks can foster informed discussions. Additionally, global cooperation between scientists, conservationists, and policymakers is necessary to develop safe and ethical gene drive applications. Conclusion. Moving forward responsibly. Gene drives hold transformative potential, but their impact on ecosystems cannot be overlooked. Through careful research, ethical considerations, and robust regulations, we can ensure that gene drive technology is used responsibly to benefit humanity without compromising environmental stability.